Hello, in this video, we're going to talk about microbial growth. So pretty much the whole video is just going to be talking about how microbes grow. And yeah, I have a picture of a petri dish because in the lab, we grow microbes all the time. Remember that when we do a 4-quadrant streak, we're trying to get colonies. And a colony is essentially a single bacteria that grew and grew and grew and created a colony. And essentially, all of the bacteria in this colony are going to be identical to each other. So we'll learn about that in this video. So when bacteria grow and divide, they divide through a process called binary fission. So they don't go through mitosis like eukaryotic cells do. So basically what a bacteria does is when it's ready to divide, it will replicate its DNA because it obviously wants the two daughter cells, if you want to call them daughter cells. So the two new cells should have a copy of DNA each. And so it's going to replicate its DNA, and then the cell just literally splits in half. It's really that simple. And so in the next slide, we'll talk about how this division happens, but essentially the cell just splits right in half, and the two new cells are going to be identical to each other, both genetically and, when the bacteria divide, they also try to divide up their ribosomes and everything else as equally as possible. And every time a cell divides, we call this a generation. So theoretically speaking, let's say we started with just one bacteria. So again, on that petri dish, if we had a single bacteria, after one generation, it will be two bacteria. And then after the second generation, now we're at four, right? Because those two are going to divide each. And then all four of them are going to divide. So now you have eight. And as you can see, because of how quickly they kind of multiply, we're going to have a lot of bacteria in just a few, hours to a day or two, depending on how fast these bacteria grow. And so that is how we get those colonies on the petri dish, because you have one bacteria that basically grows and grows and grows and divides, and then you have, a million or so bacteria on one's, location on the petri dish. And so that clump of bacteria are all coming from the same single bacteria before you started incubation. And then when you incubate, you just get millions of them. And so that is essentially how bacteria grow. So very simple, straightforward process. So here we're going to talk about exactly how the bacteria actually splits. So there is this very famous, well-known protein called FTSZ. FTSZ forms this ring that actually pinches the bacteria in half. So FTSZ, as I mentioned, is a protein. Multiple FTSZ proteins kind of assemble in this ring formation, and they will assemble in the middle of the bacterial cell. And basically what happens is the ring gets tighter and tighter. As you can see here, they're forming a ring and it gets tighter, and it basically squeezes the bacteria in the middle. And so we get this cleavage furrow, which is basically the pinching, the part where the bacteria are getting pinched. Now, at one point, the bacteria will grow a cell wall in between. So it'll make more peptidoglycan, which will create the cell wall. And so now we call this the septum because it's really dividing the two cells. And at one point, the whole, just the two cells just separate, and that's it. So pretty straightforward, as I mentioned. And as you can see, those FTSZ proteins, they disintegrate or they kind of fall apart. So you don't have the ring anymore. And so they're just hanging out now. And yeah, that's pretty much how the bacteria pinch themselves in half. Now, when we are growing bacteria in the laboratory, so specifically when we're growing them in a test tube or a flask, some kind of container, and there is limited space and limited nutrients. So this is not really the case for, if the bacteria are living outside in the environment, but if we're growing bacteria in the laboratory, the bacteria follow this growth curve. And basically, this is the number of living cells that you have over time. Now, in the beginning, when you introduce bacteria, for example, if you grab some bacteria, put them in a test tube, there's going to be this lag phase. And there's, a few hours or whatever where the bacteria are getting used to this environment. The lag phase is totally dependent on how new the environment is. So for example, if you put a bacteria in an environment that was very nutrient rich and you move them to a very nutrient poor environment, they're obviously going to be in shock. And so their lag phase will be longer. And so this is totally dependent on the situation, but there will be a lag phase where the bacteria are not really growing. They're just kind of getting used to this new environment. And then there's going to be a log phase, also known as the exponential phase, where the bacteria are now very happy and they're going to be growing and dividing exponentially. And so they're going to be doubling in number every generation. So you started with one, now there's two, and then four, 8, 16, etc. Now, at one point, again, this is if we're growing them in a test tube, they will start to run low on nutrients and they will slow down their growth. And basically, what will happen is some of the bacteria will die and some of the bacteria will continue to divide. And there's kind of like this equal rate of death and cell division. And so you'll see that the number of living bacteria kind of plateaus. And this is because the nutrients are starting to run out. Now, once there are no nutrients at all, then, and it's not just the nutrients, but also the amount of toxins and waste products that are being produced. So as the bacteria are growing, they're producing waste products. And again, we're in this limited space with limited nutrients. So once we start running out of nutrients and there's too much toxic, waste products accumulating in the test tube, then the bacteria start to actually die. And so that is the death phase in the bacterial growth curve. Now, the last thing I want to talk about is something interesting that some bacteria can do, and that is form a biofilm. So a biofilm is essentially a cluster or clump of bacteria that are growing together, and they're usually growing on the surface of something. So some kind of surface, the surface of a rock, the surface of, a person's body, but some kind of surface. And so the bacteria stick to the surface and they start to kind of colonize that surface and they will grow into this big clump. By the way, this is slightly different from what a colony looks like on a petri dish. So a biofilm is something that's happening in real life rather than in a petri dish. But anyway, so the bacteria, they clump together, they grow, and then they start to actually produce, sugars and other proteins, and they kind of secrete this, and it kind of creates this protective layer around the entire biofilm. Now, what's interesting about biofilms is that typically speaking, the bacteria that are living in a biofilm are more resistant to antibiotics and disinfectants simply because they are growing together and kind of protecting each other. And like I said, they start to produce this kind of like layer, a protective layer of proteins and lipids and carbohydrates that kind of just shields them from the outside. Now, biofilms are going to be really important in terms of hospitals and infections because in hospitals, biofilms are really annoying. So a lot of tubing and instruments that are used can form biofilms, and those are really hard to get rid of and disinfect. And so again, biofilms are a little bit annoying to deal with. By the way, in a biofilm, it's not necessarily one type of bacteria. They have discovered that some biofilms have more than one type of bacteria living in one cluster together, which is really interesting. So one example of a biofilm is the plaque that is found on your teeth. So, when you are brushing your teeth, you're supposed to be getting rid of the plaque, and plaque is essentially just biofilms of bacteria growing, on the surface of your teeth. So that's kind of why it's important to brush your teeth and get rid of those plaques before the bacteria cause any issues. So yeah, that is just what biofilms are. And I hope this video was interesting. I will see you in the next video.